D1.3 mutation and gene editing. Our two essential questions are, how do gene mutations occur, and what are the consequences of gene mutation? Now, while the DNA from which genes are made is very stable, its base sequences can be copied with great accuracy, sometimes we will have mistakes. Those random changes to the base sequence of a gene is called a gene mutation. So remember, a gene is just a sequence that makes up part of your DNA that codes for a specific protein. And so if we have a random change, we could end up with a different protein or a non-working protein, or sometimes even a protein that works better than the original. Let's look at three main types of gene mutation. Number one, substitution. This is where one base in the coding sequence of a gene is replaced by a different base. For example, adenine present at a particular point in the base sequence could be substituted by cytosine, guanine, or thymine. Base substitutions can happen by chemical changes to bases or by mispairing during DNA replication. So note here our original sequence had CGA, TAC. Now TAC codes for methionine, but we have a substitution. Instead of this thymine here, there was a random mutation, and now there is a cytosine. So the sequence reads CGA, CAC. Now CAC codes for a different amino acid. So whereas methionine would have been in the original sequence, after the base substitution, we have valine. Now, this may be harmful, this may be neutral, or this may even be positive, which is very rare, but this is what causes variation in a population that can then lead to natural selection. A second type of gene mutation is an insertion. This is where a nucleotide is inserted, so there's an extra base in the sequence of a gene. Now, this is a more major change because it requires a break to be made in the sugar phosphate backbone of the DNA molecule. And because it's an insertion, every amino acid after that point is going to be wrong because it's going to shift the reading frame. So here, note this fourth triplet in the DNA was TTG and coded for the amino acid asparagine. Note that we've inserted this C here. So now it reads TC and then the continuation here, T and G. This insertion, though, shifts the reading frame so that every amino acid after that is going to be wrong, and this can lead to a major change. It could potentially be lethal if that protein codes for an important protein that is needed for life. Next, deletion. Similarly, this is a major change. A nucleotide is removed. There is one base less in the sequence of the gene. This is going to require two breaks in the sugar phosphate backbone, one on either side of this missing nucleotide. And so here you can see TAC. We've deleted this C, which means that this A now shifts up into this position and everything behind it shifts one forward in the reading frame. So note that after this point of this deletion, we're going to have all incorrect amino acids. Now, multiple insertions and deletions can occur where two or more consecutive nucleotides are added or removed. So in summary, this is our original sequence. Here you can see a base substitution, which can lead to a different amino acid. Okay, and we have deletions and insertions, which then lead to that whole shifting of the reading frame, and therefore a very different polypeptide produced. So let's dig deeper into those consequences of base substitutions. Now, consequences are mostly neutral or deleterious, which means harmful, and in some cases, they're more than harmful, they're lethal. So in the non-coding DNA between genes on chromosomes, base substitutions would be unlikely to have any effect. Only changes to the coding sequence of genes can affect the amino acid sequences of polypeptides. So if there's a mutation over here in this non-coding DNA, we're not going to see a difference in the actual amino acid sequence of the protein produced. Same-sense mutations are base substitutions that change one codon for an amino acid into another codon for the same amino acid. This is possible because of the redundancy of the genetic code. For example, here we have ACA, which is going to code for cysteine. In this DNA, we have ACG, but both of those are going to code for codons that code for cysteine. And so we are going to have the same amino acid produced. So it's called same sense mutation, even though it's a change in the base sequence. And most likely this is going to occur potentially if there is a change in that third base, it is still going to code for the same amino acid. So there's no effect on the resulting polypeptide. Nonsense mutations change a codon that codes for an amino acid into a stop codon. Translation is therefore terminated before the polypeptide has been completed. Usually, the resulting protein does not function properly. The effects of this really depend on what the protein's function is. So this could potentially be a lethal effect if this is a protein that is required for life. So here you can see this amino acid sequence has been cut off. This stop codon was produced. So the ribosome complex at this point would dissociate and we would have a shortened polypeptide chain depending at which point along the DNA this mutation occurred. Missense mutations alter one amino acid in the sequence of amino acids in the polypeptide. Now, they may not have much effect if the new amino acid has a similar structure and chemical properties to the original one. And so if an amino acid that was, say, positively charged, hydrophilic, was replaced by another one that was also positively charged, hydrophilic, it may not have a large effect on the function of the resulting protein. However, we may see that they could have severe effects and even lethal effects, like we saw with sickle cell disease, where we had a single base substitution that resulted in a missense mutation. One amino acid was changed and it led to sickle cell disease. In fact, many genetic diseases are due to missense mutations. Now, a very small proportion of missense mutations do actually improve the function of a protein and increase the individual's chance of survival. These beneficial mutations, though, are massively outnumbered by deleterious mutations or harmful ones, but they're arguably more significant because this is what leads to evolution and adaptation, are mutations that happen to be beneficial. So when a base substitution mutation happens in one individual in a population and then is inherited by offspring, a new allele of a gene has been produced and the genetic diversity of the population increases very slightly. So remember, an allele is a form of a gene. So new forms of the gene arise through random mutation. Now, in evolutionary terms, this is beneficial for the population as a whole. Variation in a population is critical because if there's some kind of environmental change, there's a higher likelihood that some members of the population will be able to resist that change and survive, and therefore it doesn't wipe out the whole population and the population can continue. Single nucleotide polymorphisms, or SNPs, are the result of base substitution mutations. And because of the degeneracy of the genetic code, how they may have more than one codon that codes for a single amino acid, they may or may not change a single amino acid. So the presence of some SNPs is associated with certain diseases. These correlations allow scientists to look for SNPs to determine an individual's genetic predisposition to develop a disease. So we look for these single locations where we know there's differences between members of a species, some of which may show a genetic predisposition to a certain type of disease. Insertions and deletions are less likely than substitutions to have beneficial consequences. Remember, we're seeing an entire shift in the reading frame when we either insert or delete a nucleotide. Major insertions or deletions almost always result in that coded for polypeptide ceasing to function at all. Now, minor insertions and deletions of one or two nucleotides can also result in total loss of function of a polypeptide because they are frame shift mutations. They change the reading frame for every codon from that mutation onwards in the direction of transcription and translation. So if everything shifts over one letter because we have a deletion, you can see that's going to change every codon after that fact. Now, insertions and deletions in a multiple of 3 are not considered frame shift mutations because we're basically just going to have one, say, less amino acid. Everything after that will still be read correctly. So it's not shifting the whole reading frame. It's only going to be then affecting that one missing or extra amino acid if there's a multiple of 3. It can still have severe consequences. Having that one extra or one too few amino acids, it could cause radical changes in the structure of the protein and therefore affect whether or not it can carry out its functions. So, what are the causes of gene mutation? Gene mutations can happen at any time, but the chance is normally very low as DNA is resistant to chemical change. Remember, it is very stable. There is an increased risk, though, during DNA replication. Base pairing errors can sometimes be made during this process, and sometimes they don't get corrected by DNA repair. The frequency of mutation is then increased by external agents known as mutagens. So mutagens increase the frequency of mutations. And there are two types, radiation and chemicals. First, radiation increases the mutation rate if it has enough energy to cause chemical changes in DNA. Let's review the electromagnetic spectrum. Here we have visible light, 400 to 700 nanometers. Remember that as we go towards the blue end, we're seeing these shorter wavelengths, which are higher energy. And so if we go past visible light, we're going to get to ultraviolet or UV light. You may know that UV light is associated with skin cancer. And it's that property, that radiation of UV light, because it is even shorter wavelength, it's higher energy, and it can lead to an increased chance of mutations, which then lead to skin cancer. Even beyond that, we go to X-rays. This is why when you go to the dentist, you put that big heavy smock on you because it's protecting you from excess X-rays. Beyond that, we have gamma rays, cosmic rays. We're just getting shorter and shorter wavelength, meaning higher and higher energy. And so we're going to see that these are mutagens. Gamma rays, X-rays, alpha particles from radioactive elements such as radon are mutagenic. Shortwave ultraviolet radiation in sunlight is also mutagenic. That high energy shortwave radiation can lead to a higher rate of mutations. Number two, some chemical substances can cause chemical changes in DNA, so therefore they are mutagenic. Examples are polycyclic aromatic hydrocarbons and nitrosamines found in tobacco smoke. Mustard gas is also mutagenic and was used as a chemical weapon in the First World War. Randomness in mutation. Mutations are described as random changes. Whether or not anything can be truly random is a debatable question, but mutations certainly are unpredictable. They can't be directed by living organisms to achieve an intended outcome. The consequences of a mutation have no influence on the probability that the mutation will or will not occur. There's no natural mechanism for changing a particular base with the intention of making a beneficial change to a trait that has been discovered or is likely to ever be discovered in living organisms. They are random. Mutations can occur anywhere in the base sequence of a genome, although some bases do have a higher probability of mutating than others. This is because mutation is a chemical change, and some chemical changes just happen more easily than others. The position of a base within the genome also affects the chance of mutation. This is because the differences in how coding and non-coding DNA sequences are used then affects the likelihood of whether that mutation is going to occur. So mutation is random, and a consequence of randomness is that a mutation is unlikely to be beneficial. Genes have developed by evolution over long periods of time, in some cases over hundreds of millions of years, so a random change is usually neutral or harmful. To the best of our knowledge, there is no mechanism for testing out mutations acquired during the lifetime of an organism so that only the beneficial ones are passed to offspring. Let's distinguish between the consequences of mutation in germ cells and somatic cells. Now, a germ cell is a cell that gives rise to gametes. Those are those eggs, sperm, those sex cells that then form the offspring. So when you die, the genes in germ cells can be passed to offspring. Those mutations are going to be inheritable. So a new allele produced by mutation in a germ cell can be inherited. In rare cases, a new allele could confer an advantage, but in far more cases, the mutation causes a genetic disease. It is therefore particularly important to minimize the number of mutations in gamete-producing cells within the gonads, which are the ovaries and testes. Okay, but it's very different if we're in a somatic cell, which is the body cells, any other cell besides the sex cells. Any mutations in the body cells are eliminated when an individual dies. So there's limited consequences. For example, if UV radiation did cause cancer due to a mutation in a skin cell, the offspring will not be born with that mutation. Because the mutation happened in a body cell, it is not inheritable. So the consequences of mutations in the genes that have roles in control of the cell cycle and cell division can be much greater. Remember that cancer is where the cell cycle is just happening out of control and the cells are just dividing, and that's what forms a tumor, uncontrolled cell division. And this can happen if a mutation occurs in proto-oncogenes, because mutations then change them into oncogenes, which are cancer-causing genes. If you think in a hospital, the cancer treatment department is called the oncology department. So onco is referring to cancer. And so mutation is our source of genetic variation. Any kind of question you see on the IB exam about evolution or natural selection, the first mark is almost always just establishing that variation in a population exists as a result of random mutation. And so here we're tying that in with some of these previous units we've looked at. Here we're seeing mutation is the source of genetic variation in a population. So just remember that as a hint. When you're answering any question about natural selection, just start by saying variation in a population arises due to random mutation. So an allele is a variant of a gene. It differs in one or more bases from other alleles of the gene. Mutation changes the base sequence of the gene, so it changes one allele into another. So alleles show up due to random mutation. We have just a mistake in that base sequence. Mutation increases the number of alleles in the population, so it increases genetic variation. Meiosis and sexual reproduction can increase variation by generating new combination of alleles, but the original source of all genetic variation is mutation. Again, most mutations are either harmful or neutral, but nonetheless, mutation is needed in all species because natural selection requires genetic variation. Species can't evolve without it. If you have members of a species that all have the exact same genes, then one environmental change comes along and it wipes out the whole population. In order for natural selection to happen, we're going to need some individuals to have a mutation that provides some sort of advantage, that they're then going to have a higher reproductive capability. They're going to be able to pass on their genes to make more offspring, and those genes then have a higher frequency in the population. And that's what natural selection is. So especially during times of rapid environmental change, populations must adapt to new conditions. Of the many alleles produced by mutation, only a small proportion will confer characteristics needed for this adaptation. Without mutation, genetic variation in a population would decline over time. This would make natural selection impossible. If we don't have variation, natural selection cannot occur. This inevitably leads to extinction when the environment changes and the population is not able to adapt and evolve. So mutations are really, really important because they allow for natural selection to occur, those rare beneficial mutations. Quick nature of science moment. Commercial genetic tests can yield information about potential future health and disease risk. One possible impact is that without expert interpretation, this information could be problematic. So think about it. If you could go to some kind of drugstore and buy a genetic test that tells you whether you have a predisposition for a certain disease, that might be really helpful, because then maybe you go in and you get extra testing more regularly through your life because you know you have a higher probability of developing this disease. But what if you decided, well, I have a higher probability of getting this disease, and you end up just giving up and you say, well, I'm not going to work hard in school then, or I'm not going to try to get a good job or go to college because I'm just probably going to get this disease. And so it's really, really important that when genetic testing of any kind is carried out, that there's an element of counseling that goes along with it. And so commercially, being able to buy a genetic test at a drugstore could potentially be problematic if there's no counseling that is coming along with interpreting the results. All right, this is the end of the SL content. If you're an SL student, you're good to go. HL students, we have just a bit more. Here we go. First up, we have all this DNA containing all these A's and T's and C's and G's. How do we know what a particular sequence, a gene, actually does? The way we figure that out is a technique called gene knockout. It allows us to investigate the function of a gene by changing it to make it inoperative. We knock it out so then we can see, well, what's missing from the organism, and then we know what that gene actually does. So it's a technique for discovering the function of genes. It's carried out in different model organisms, usually mice. And here are the stages of gene knockout in mice. First up, DNA is prepared with a base sequence that allows it to be inserted into the genome of embryonic mouse cells as a replacement for a target gene, which is therefore deleted. So the first thing we're going to do is we're going to delete a gene, put in some known DNA that doesn't do whatever this deleted sequence does. Cells where this procedure have been successful are then selected. They're grown into adult mice. Now, instead of having 2 copies of the target gene, these adults will only have 1 copy. The other copy has been deleted in step 1. So then males and females that only have 1 copy are mated, and we'll learn about this in our inheritance unit. 25% then of their offspring can be expected to have no copies of the target gene. And these are our knockout mice that we're going to study. The phenotype of these knockout mice is then investigated to find out which traits have been changed by deletion of the target gene. So we're taking a mouse, we're deleting that gene, and then we're seeing, well, what are they missing? And that helps us to understand what the gene does. In mice, several thousand knockout strains have been developed, each lacking 1 gene, and these strains have been widely used in research. For example, piezo-2 knockout mice were found to urinate less frequently than normal individuals. The piezo-2 gene has been found to code for a mechanosensitive ion channel that acts as a pressure sensor in the bladder. Humans who naturally lack a functional piezo-2 gene also show impaired bladder control. So by studying these mice and figuring out what this gene does, we can now use this to develop medical treatments now that we understand the genetic basis of that condition. In a few model organisms, such as yeast, there are now libraries of knockout organisms for all the genes in the genome. Quick note, you do not need to know details of the techniques, but you should appreciate that a library of knockout organisms is available for some species used as models in research. Next up, CRISPR. Use of CRISPR sequences and the enzyme Cas9 in gene editing. Now, CRISPR-Cas9 gene editing is based on a natural system that exists in many species of prokaryotes. The 2 main elements are CRISPR regions within the genome and the enzyme Cas9. Now, you are not required to know the role of the CRISPR-Cas system in prokaryotes, but you should be familiar with an example of the successful use of this technology. So, just an overview. This is a naturally occurring enzyme that bacteria, prokaryotes, use to try to identify non-self DNA and destroy it. So what they're going to do is this enzyme, Cas9, is going to move along a DNA sequence. It has some guide RNA here that when it forms complementary base pairing with a specific target sequence on the DNA, it then identifies that target sequence as non-self. Maybe it belongs to a virus that's trying to attack the bacteria. And so Cas9 is a naturally occurring defense tool. And so what it does when it identifies that sequence through complementary base pairing, it cuts that DNA. 2 cuts, 1 for each strand, and therefore breaks that DNA so that if it was, say, viral DNA, that viral DNA would no longer be able to do transcription and translation and make those proteins that the virus wants to make in order to be able to replicate and infect other cells. So we're going to see that we can use Cas9 enzyme to identify a specific location in DNA, cut it, and then take the desired DNA sequence that's added to repair. And in this way, we are cutting and pasting portions of the genome, cutting and pasting DNA so that we can move genes in between organisms, even in between species, because the genetic code is universal. So CRISPR, this is an acronym for clustered, regularly interspaced, short palindromic repeats. So the repeats is because the same base sequence occurs several times in the genome. Here you can see all of these R's representing repeated sequences, and this is color-coded in brown. So you can see here these repeated sequences starting with GTTC. Here it is again in brown, GTTC, representing this one, right? G, T, T, C, etc., representing this one. So these brown ones are these repeated sequences. They're short. The number of base pairs in the repeat is between 23 and 47, so pretty short. They're clustered. The repeats are going to be grouped in one part of the genome, and they are regularly interspersed. The repeats are separated by other base sequences of similar length called spacers. And here you can see they're color-coded here. You can see an example. It's just a different DNA sequence. Now, each spacer is unique. There can be as few as 2 or over 120 in a CRISPR locus. They are derived from viruses that infect prokaryotes, and they allow for viral DNA sequences to be recognized if infection occurs, and then cut and destroyed. Palindromic. Each repeated sequence have parts which read the same backwards as forwards. It's not truly palindromic, but the name CRISPR containing the letter P has stuck. CRISPR doesn't have quite the same ring to it. So, prokaryotes make guide DNA by transcribing a repeat and an adjacent spacer. The repeat is then used to bind to Cas9, and the spacer is going to be what searches for those viral base sequences in DNA. Cas9 is this enzyme. It looks like kind of a grayish cloud here. It can find those specific DNA base sequences using that guide RNA that is bound to it. The spacer on the guide RNA forms a variable 17 to 20 base sequences at the 5' end of the guide RNA. It is complementary to the target DNA that is being searched for. The repeat forms other parts of the guide RNA, which are partly double-stranded, generating loops and a distinctive molecular shape that promotes binding to the Cas9 enzyme. So Cas9 moves along the DNA molecules. It uncoils them by splitting these complementary base pairs. It brings the DNA adjacent to that variable base sequence of the guide RNA. So it runs this guide RNA along the DNA to look for that complementary sequence. Within its structure, Cas9 contains 2 endonucleases. If the target sequence is recognized, the endonucleases cut 1 sugar phosphate bond in each of the strands. So then a double strand break is the result in the target DNA. And this is how prokaryotes recognize and destroy foreign DNA within their cells, especially viral DNA. We can use Cas9 CRISPR in gene editing. Gene editing has long been an aspiration of molecular biologists. If it could be done reliably, specific changes could be made to organisms such as crop plants. It would also allow genetic diseases to be eliminated from human populations. For example, we could take a gene for pest resistance and we could insert it into a corn plant. We would no longer need to spray that corn with all of our chemical pesticides if there's a gene in it that allows it to be pest resistant. We could cut and paste the sequence of the DNA that would lead to a genetic disease and therefore fix that mistake and eliminate genetic diseases from the population. So these are 2 specific examples of how Cas9 CRISPR could be used in gene editing. Now, gene editing requires a method for ending a base sequence in the genome, that is the target, which is what we just talked about, how that works to cut a specific sequence in the DNA, but then it also has to be replaced by the desired sequence. This is known as search and replace. The Cas9 CRISPR system can be used to end a target sequence, and molecular biologists have modified it so it can make changes to the sequence. Now, quick nature science moment. Certain potential uses of CRISPR raise ethical issues that must be addressed before implementation. A lot of people find that this really crosses the line with, are you, quote, playing God? You're copying and pasting genetic code, and most people would agree that this seems like a good thing to be able to eliminate genetic diseases from a human population, but what if it's taken further? There's a gray area with ethics. At what point is it wrong? What if you are fixing a genetic disease so that a child is no longer going to grow up with type 1 diabetes? This seems like a great thing, but is it ethical to edit the DNA to prevent a child from having a learning disability? Is it ethical to edit DNA to change someone's physical appearance? Maybe to prevent them from having a receding hairline in their 20s? At what point is this just playing God rather than trying to solve problems that affect people's quality of life? There are different regulatory systems around the world, And so right now there's an international effort to try to harmonize regulation so we all have the same rules about the application of genome editing technologies such as CRISPR, so that we're all following these same guidelines and making sure that we're making ethical decisions. Lastly, hypotheses to account for conserved or highly conserved sequences in genes. So first, conserved sequences are identical or nearly identical across a species or group of species. Highly conserved sequences are identical or similar over long periods of evolution. So these may occur across an even wider range of species. Here you can see histone protein. Remember that histone proteins are the proteins that DNA is wrapped around twice a cluster of 8 histones in order to pack it tightly into the nucleus. And they're also then going to play a role in which parts of the DNA actually end up being made into proteins through transcription and translation. Because this is a key protein for packaging up that DNA, we're going to see that it exists in many eukaryotic species. Here are 5 examples. Now, each of these letters represents a different amino acid. So these are, of course, coded for by DNA. Now, note that the first 3 amino acids in each of these 5 species are identical, but the amino acid at the 4th position is going to be the same for humans and chimps, but different for mice, rats, and cows. But you'll see that these amino acids are almost the same in all of these species. So this would be an example of a conserved sequence. So there are 2 hypotheses for why sequences are conserved. First, functional requirements for gene products. Many conserved sequences are in protein coding elements of the genome or in elements that are transcribed to make ribosomal or transfer RNA or sequences used to regulate gene expression. So these are all functional sequences. Either they code directly for a protein or they're critical and they code for something else that is needed for life to exist. So these are all elements in the genome that have that obvious function. If their function remains unchanged, there might be little or no change in the base sequences over long periods of evolutionary time. Like we saw with that histone protein, it's so functional that we're not going to see big differences because it plays such an important role. It's going to be really similar throughout all of those different species that were looked at. Non-coding elements are also conserved in the genomes of many organisms, the functions of which are largely unknown, but the fact that they're conserved, sometimes across a wide range of organisms, suggests they do have functions. We just haven't figured out what they are yet. So maybe that's something that you're going to figure out in the future, is what are some of these mysterious DNA sequences actually doing? And then lastly, sequences may be conserved just because they have slower rates of mutation in that portion of the genome. Note that mutation rates are not identical throughout the genome. The conserved non-coding elements might be in regions of the genome where mutation rates are just lower. All right, that brings us to the end of D1.3. Good luck out there.